Welcome everyone. Today we are going to learn about membrane fluidity. In 1970, scientists proposed fluid mosaic model of plasma membrane. The term fluidity is because of the mobility of lipids and proteins that are present in the plasma membrane. So both lipid component and the protein components are mobile in the plasma membrane. In my previous video, I have discussed about the mobility of lipid molecules. I will take a separate class on protein mobility also. Now in that video, I have mentioned that lipid molecules perform 4 different movements. One is rotation, flexion, lateral diffusion and flip-flop. And this is for this movement the plasma membrane is mobile or actually fluid in nature. So today's topic is membrane fluidity. Membrane fluidity. The fluidity of the membrane has to be regulated very precisely because certain membrane functions cease when we increase the membrane viscosity beyond a threshold level by experimentally. Now viscosity is the measurement of resistance to flow. That means if we make the lipid bilayer less fluid, then some certain functions of the membrane will stop. So fluidity is very important. Now there are 4 factors responsible for this membrane fluidity. I will write here. The first is temperature. Second is the length of fatty acid chains. Third factor is degree of saturation of the same fatty acid chains. And fourth point we have to remember is that lipid composition. So these are the 4 factors that control the membrane fluidity. At first I will talk about temperature. So as we know that membrane is lipid in nature, we can take an example like butter. If we heat a butter, the butter actually melts, so it becomes fluid. The same is true, and if it cool down, then it becomes solid. So the same is applicable for the plasma membrane also. At physiological temperature, that means at 37 degrees centigrade, the membranes are actually fluid in consistency, so it is fluidic in nature. But what happens when we cool this membrane? So let me point down. If we cool the membrane below 37 degrees centigrade, one very important phenomena occurs, that is called phase transitions. What happens? The membrane that basically liquid in nature, because at 37 degree the membrane is liquid in nature, fluid in nature. So the liquid state of the membrane undergoes into a gel-like state or semi-solid state, gel state. So this is called phase transitions. Now when it becomes gel state, that means it becomes less fluidic in nature. So less fluid. I can write less fluid. Why it becomes less fluid in nature? The answer is here. Below the phase transition temperature, let's suppose 37 degrees is the phase transition temperature. Below the phase transition temperature, the rate of lateral diffusion drops very precipitously. So the mobility of the lipids actually drops. That's why it becomes less fluid in nature. Now the opposite will be true. If we warm the membrane greater than 37 degrees centigrade, what will happen? It becomes more fluid. So it will become more fluid. Okay. Now this temperature or the phase transition temperature actually depends on also these 2 factors, length of the fatty acids and degree of saturations. as I have mentioned, these are the 4 factors that control the fluidity. Temperature also control the fluidity, and these 2 factors also control the temperature. How it occurs? Let me explain. Let's suppose, here I am explaining. Let's suppose there is 2 type of lipid molecules present in the membrane. This is the head group of the lipid, and let's suppose the fatty acid chains here look like this, and there is another type of lipid molecules present that has the same polar head group, but the fatty acid chain is here, look like this. Okay, the same polar head group. Let's suppose this is the example of one phosphoglyceride like phosphatidylcholine. Both are phosphatidylcholine. One phosphatidylcholine phosphoglycerides contain this lipids contain long and saturated fatty acids, and this phosphatidyl contains short and unsaturated fatty acids. Let's take an example. So long fatty acids means these fatty acids contain more carbon atom. So like a carbon fatty acids could be C20, a fatty acid could be C14 also. So the number of the carbon molecules in the fatty acids actually tells the length of the fatty acid. So if it is long, then more carbon atoms are present, and if it is short, then less carbon atoms is present. Now what is the significance of presence of long fatty acids beside short fatty acids? We know that long fatty acids can make more van der Waals interactions. So for long fatty acids, it can make more van der Waals interactions. Sorry, the spelling will be van der Waals interactions. So it will make more van der Waals interaction than the short fatty acid chains because it has less surface area. And when long fatty acid chains makes more van der Waals interaction with its neighbors, it tightly pack with each other. So it will assemble or aggregate tightly with its neighbor fatty acids. So if there is another lipid molecule present, they will tightly pack. And also you see here saturated fatty acids. Saturated fatty acid means those fatty acids does not have carbon-carbon double bonds, and unsaturated means those fatty acids contain carbon-carbon double bonds. Now in my previous video I have mentioned that saturated fatty acids are straight in structure, they are straight, and in case of unsaturated fatty acids, if there is any carbon-carbon double bond present in the fatty acids, where the carbon-carbon double bond is present, there will be a kink or a bend in the structure. Now straight fatty acids can make stable van der Waals interactions. So for saturated fatty acids, they make stable van der Waals interactions. More stable van der Waals interactions because they are straight. They can make van der Waals interactions with the neighbor fatty acids, more stable van der Waals interactions. But for unsaturated fatty acids, there will be less stable van der Waals interaction, and for short fatty acids, there will be less van der Waals interactions. So for these 2 reasons, long saturated fatty acid containing lipids in the plasma membrane will make more. So this type of fatty acid will make, or lipids make, more ordered gel-like state or gel state. Okay, so this type of lipids will make more ordered gel state. Now I will show you here. Let's suppose these lipid molecules, there is more lipid molecules. Let's suppose I'm giving four examples. Let's suppose the lipid bilayer of the plasma membrane is made up of this type of fatty acid containing lipids. So this will look like this. So this is one layer of the fatty acids, and let's suppose this is the another layer of the fatty acids. So if the bilayer of the lipid plasma membrane contains this type of long and saturated fatty acid containing lipids, then it is actually more ordered gel states. Now if we warm or put heat on this bilayer, so heat, what will happen here? As you can see, the fatty acids that for long and saturated fatty acids of those lipids actually make little overlap. So there is little overlap, there is not overlapping, that's why it is very ordered. So there is little overlapping, there is little overlapping between the fatty acids. But when heat actually we hit this bilayer structure, what will happen? These fatty acids or the hydrophobic fatty acids will actually becomes disordered. So disorder will be introduced. So heat actually introduced disorder, and for this disorder, what will happen? These lipid molecules now look like this. How it will looks, I will show you. Now the fatty acids of these lipid molecules will overlap significantly. Now this fatty acid will overlap significantly, and that will decrease the thickness of the membrane also. Let's remember this. So the thickness of the membrane will decrease. Lipid molecules with long and saturated fatty acids, it will make more ordered gel-like state. When we heat it, it will the heat actually generates disorder, and it becomes fluid in nature. So gel to liquid state, it will convert it. So more fluid, it becomes more fluid. But simultaneously, as well as when we heat this type of lipids in the bilayer, it will actually decrease the thickness of the membrane also. This is the fact. Now let me explain how these two factors actually determine the phase transition temperature. Third point is that long fatty acids containing membranes are less fluid because they make more van der Waals interactions, and that will actually helps them to tightly aggregate with each other, so it makes less fluid. Fourth point, we can say that short fatty acid chain containing lipids in the plasma membrane or membranes make it more fluid. Another point we have covered, that the fifth is saturated fatty acids make stable van der Waals interactions that will make it less fluid, and unsaturated fatty acids, and also degree of saturation means how many carbon-carbon double bonds are present. As many as carbon-carbon double bonds will be present, it becomes more fluid. It becomes more fluid. Okay, so we have covered up to this. Now how these two factors actually, or the saturation factors, actually control temperature, let's see. Scientists have performed one experiment where they have made artificial lipid bilayer. Artificial lipid bilayer membrane are called liposomes. All the membrane research actually performed on liposomes. Okay, I have already described the structure of liposome. I am not describing it here. So scientists performed an experiment. They made an artificial lipid bilayer membrane that is called liposomes to observe the membrane fluidity or the the membrane fluidity. Now they have taken here cholesterol and two kinds of phosphatidylcholine. One phosphatidylcholine that contains saturated fatty acid, and another type of phosphatidylcholine that contains unsaturated fatty acids. Okay. Now they observe that when they mix when they mix cholesterol with these two kinds of phosphatidylcholine, this saturated phosphatidylcholine actually segregate, they actually clump, they segregate to form more ordered liquid phase. Now I have mentioned that there is two state, that is liquid state and gel state, that is called phase transitions. But now this gel state are actually can be generated with long and saturated fatty acids, as I have already mentioned, long and saturated fatty acids. If the lipids contain long and saturated fatty, it will actually make a highly ordered gel-like state. But if you only take this lipid molecules like phosphatidylcholine only with saturated fatty acids, let's suppose this is not a long fatty acids, only saturated fatty acids, then it will not make a gel state, but it will make a ordered liquid phase. Okay, you have to understand, liquid phase is another one phase, gel phase is another phase, but it will make with PC saturated yes, I am writing here, with saturated PC, it will make more ordered liquid phase. And if we take this PC that is unsaturated PC will make less ordered liquid phase. Now they have observed that in case of more ordered liquid phase, the melting temperature means simply the temperature at which the gel state of this membrane will melt, so it becomes gel state to liquid state. So the melting temperature is high here, but in case of less ordered liquid phase, the melting temperature is low. If there is phosphatidylcholine with saturated fatty acids, that will make stable van der Waals interactions, that will make more stable, they will aggregate with each other tightly. And we have mentioned that saturated fatty acids make it less fluid, so it is less fluid. To melt it, it will require more temperature, more heat. So as it is less fluidic, so to make it fluidic, it will require more heat. But in case of unsaturated phosphatidylcholine, it makes less ordered liquid phase. That means unsaturated, it means it is more fluid already, so it will require less heat for the melting. So it requires melting the melting temperature is lower in case. Now let's see how melting temperature differs with the degree of saturations. So we will measure the melting temperature of common carbon 18 fatty acids. Now at first we will write the name, then the number of carbon cis double bond, and then we will measure the melting temperature at degree centigrade. Okay, so first example of carbon 18 I can write here is stearic acid. Stearic acid is an example of carbon 18 fatty acids with no carbon double bond, so 0. That means it is saturated fatty acids. The melting temperature is high, 70 degrees centigrade. As I mentioned that saturated fatty acids is less fluidic nature, so it requires more heat, more temperature for to melt the fatty acids. Then one example of fatty acid with C18 carbon, the length of this fatty acid is same, like oleic acid. This fatty acid contains one single carbon-carbon cis double bond, so it is monounsaturated fatty acid. It is unsaturated but monounsaturated fatty acid. Now let's see how the melting temperature is for this oleic acid. It drops from 70 degree to 13 degree, so that means at least 57 degrees centigrade is lower in this oleic acid. That means just by introduction of one single carbon-carbon cis double bonds, that means it becomes unsaturated, so it becomes more fluid, so it requires less heat. How much? 57 degree difference. Okay, but the length of the fatty acid is same for C18. Now let's take example another. Example third is linoleic acid. This fatty acids contain two cis double bonds. Now the temperature also fall here, minus 9 degree centigrade. Okay, minus 9. So it also falls from here to approximately 22 degree. You see. Another is example is like linolenic acid. All these acids are C18 fatty acids. Now linoleic acid also have three cis double bonds, so these two are examples are polyunsaturated fatty acids, and the temperature drops here from 9 degree to 17 degree centigrade. I'm also giving one example of fatty acids called eicosa pentanoic acid. In short, we call it EPA. This is one example of omega-3 fatty acids, but obviously these fatty acids contain carbon number of 20. This is somehow it is longer, but this fatty acids contains 5 carbon-carbon double bonds, and as you will surprised to see the melting temperature, it is minus 54 degrees centigrade. So you see here, as much as the number of the degree of saturation increases, the melting temperature is actually decreases because it becomes more fluid in nature. So to melt it requires less energy or less heat. Also, it is a higher or longer fatty acid chains. Now let's talk about the fourth factors, that is the lipid composition. How lipid composition of the plasma membrane actually regulate the membrane fluidity. Now let's talk about how lipid composition affects the membrane fluidity. You can remember that almost all sphingomyelin and phosphatidylcholine. Now these two lipid molecules falls under two different category of lipids present in the plasma membrane. I have mentioned in my previous video that in the plasma membrane there are three major categories of lipid molecules are present. The first is phosphoglycerides, second is sphingolipids, and third is cholesterol. Phosphatidylcholine falls under the phosphoglycerides and sphingomyelin falls under the sphingolipid lipid molecules. So almost all sphingomyelin and phosphatidylcholine are present in exoplasmic leaflet. If you have watched my previous video, you know that there is two layers of lipid molecules present in the plasma membrane. The outer layer is called exoplasmic leaflet and inner layer is called cytosolic leaflet. So almost all sphingomyelin and phosphatidylcholine are present in the exoplasmic leaflet, that means the outer leaflet, and this make the outer layer of the plasma membrane less fluid. Why? Because for sphingomyelin it is sure that the fatty acid chain present in the sphingomyelin is long and more saturated than other phosphoglycerides. That's why it can make extensive van der Waals interactions with the neighbor lipid molecules. So it makes the lipid bilayer or lipid layer, especially exoplasmic leaflet, less fluidic. Now another point we can remember about lipid composition is that phosphatidylethylamine, phosphatidylserine, and phosphatidylinositol. Now these three phosphoglycerides are preferentially located, remember the term, preferentially located in the cytosolic leaflet. leaflet, and this lipid molecules makes the inner layer or the salicylic leaflet more fluid in nature. Okay. Now another point you can remember, that high concentration of cholesterol. Now the high concentration of cholesterol is actually present in the natural plasma membrane. So natural plasma membrane contain high concentration of cholesterol. Now cholesterol has steroid rings. Intercalation of the steroid rings with the fatty acids tends to immobilize the lipid molecules. That's why it makes the high concentration of cholesterol makes less fluid membrane. So it makes less fluid membrane. Because of the presence of steroid ring, it intercalates with the fatty acids and it tends to immobilize the lipid molecules, so makes less fluid plasma membrane. Another point we can remember, that low concentration of cholesterol, what will happen? Then it becomes more fluid in nature, because opposite will happen. One point we also can remember for the cholesterol, that cholesterol acts as a buffer. As I have mentioned, that if you warm the plasma membrane above the phase transition temperature, like greater than 37 degrees centigrade, the membrane becomes more fluid. Now if cholesterol is present in the membrane, then what will happen? Cholesterol will try to decrease the fluidity, as because it is, if we warm it more than 37 degrees, the membrane become more fluid, so cholesterol try to decrease the fluidity. Also in case of cooling, if we cool down the membrane below 37 degrees centigrade, then it becomes less fluid, so cholesterol also then try to increase the fluidity, so it try to increase. So here cholesterol actually acts as a buffer. Also one point you can remember, that is, I have mentioned previously, that plasma membrane go through two phase. This is called phase transition. One is called gel phase and another is called liquid phase. Now I have mentioned that gel phase is highly ordered, okay. But the liquid phase also has two category. One is more ordered liquid phase, and one liquid phase is called less ordered liquid phase. I have already mentioned here. Now if I compare with gel phase with this more ordered liquid phase and less ordered liquid phase, it will like this, that gel phase is more ordered than the more ordered liquid phase than the less ordered liquid phase. Now cholesterol has opposite effects on the gel phase and liquid phase. Above the phase transition temperature, that means above the 37 degrees centigrade, cholesterol favors this phase, cholesterol favors more ordered liquid phase, okay. But it cholesterol disrupt the gel phase, okay. So what I am talking about, more than above the phase transition temperature, that means above 37 degrees centigrade, what will happen? The gel phase will convert it into or undergo a phase transition, it becomes liquid phase. Now presence of the cholesterol, it try to make the membrane in the middle of the ordered situations. It will not actually it will prevent the membrane from undergo gel phase to less ordered liquid phase. It will try to bring the membrane to more ordered liquid phase than the less ordered liquid phase, but it will disrupt the gel phase. Hope this video will help you. Thank you for watching and always keep learning.